Hello student, I'm Mr. Leung. So in this video, I'm going to talk about the chapter 3 assignment. So just like before, it will be divided into two parts. For the first part, it will be the MC questions. And the second part will be the long questions. So before you do the assignment, please take a look at the book or your notebook, okay? So to get familiar the concept of cellular transport, the cell membrane structure, the functions, okay? And also the 4 types of cellular transport: diffusion, osmosis, active transport, and phagocytosis. Okay, take a look first and then you can do the assignment. So firstly, we are going to talk about question 2. So for question 2 is talking about the cell membrane. So there are 2 pathways, okay, to show that for the substance to move across the cell membrane. For the X, okay, X pathway is talking about the substance. It can pass through the phospholipid bilayer directly. And for Y is that it can be transported by the protein molecules, for example, the channel protein or the carrier protein that we mentioned in the lessons. Okay, so for the questions, it's talking about vitamin A and calcium are nutrients in food. We need them. Okay, so for vitamin A, it is a non-polar substance, and calcium ion is an inorganic ion. So for this part, I would like to mention that vitamin A, which is fat soluble, so that's why it is a non-polar substance. And for the calcium ion, because it has 2 positive charge, so that's why it is a polar substance. So that's why at least you need to know the nature of the substance first. And then you can determine which way it should pass through, move across the cell membrane. And we call the very basic concept of the cell membrane, the arrangement, and also how can it be differentially permeable, because the basic idea of polar likes polar and non-polar likes non-polar. For example, for the phospholipid bilayer, okay, for this phospholipid molecule, so for the head, which is a phosphate group, so and for the tail, they are the fatty acid chain. So which part is polar, which part is non-polar? So you should be able to recall it from the book. So after you know which part is polar and which part is non-polar, and then you will know that for the vitamin A and for the calcium ion, which substance they can move across directly, pass through directly the cell membrane, the phospholipid bilayer, or they need the protein molecules to carry them across the cell membrane. So that's for vitamin A and calcium. And one more bonus mark I would like to give you is that if you can tell me vitamin C, it should move across the cell membrane through path X or path Y, okay, I will give you one more bonus mark. So that's question 2. And for question 3 is talking about the experiment of osmosis. And what we are going to prepare is 3 fresh potato cylinder with different length, okay, and before and after they are immersed into different solution, okay, in 3 sucrose solution, that is a sugar, okay, sugar solution of different concentration for 1 hour. So what will be the final length, okay, of the potato cylinder? So because it is a experiment, so that's why I grabbed this chance to recall the basic concept of the chapter 1, IV, DV, and CV. For IV, that's something we are going to study, okay, so and that is also something we are going to change. So that's the sucrose solution of different concentration. We change it X and Y, different concentration. And then for the DV, that means it will change, depends on the IV. That's something we are going to observe. .dv, okay, that's something we are going to observe. So that's why we are going to observe the change of the length of the fresh potato cylinders. So something for example, for the potato cylinder, we put it in solution X, it becomes longer. In solution Y, it becomes shorter, and in solution Z, okay, it remains unchanged. So that's the DV we are going to observe. And for the CV, control variable, that's something we need to keep constant in different setup. We have 3 setup, solution X, Y, and Z. And in these 3 setups, we need to keep it as a fair test. So that's why, for example, something we need to we need to keep it constant. For example, the initial length of the fresh potato cylinder. Okay, we cannot use, for solution X, I use the initial length longer, okay, but Y is shorter, something like that. No, okay, we should maintain them as the same length, same initial length. and also the volume of sucrose solution. Okay, so that's a reminder for chapter 1. So go back to the question. After we get the result, okay, longer, shorter, same. Okay, so we need to arrange the water potential, okay, from the lowest to the highest, okay, as the order. So what will be the sequence? Okay, so how can you do it? Is that we call the basic concept of water potential. The higher the concentration of the solution, the lower the water potential of the solution is. So that's why if the concentration, for example, 10% sucrose solution, okay, the water potential is lower than that of the 5% sucrose solution. Okay, so that's the basic concept here. And then, but in this question, we do not know the concentration, but it doesn't matter. We can still try to determine, okay, the water potential by what? By using the result. Okay, the fresh potato cylinder, it become longer, shorter, or remain unchanged. So we can determine the direction of the net movement of water molecules based on the change of the length of the fresh potato cylinder, because it can help us to determine the water potential of the sucrose solution. I give you one example first. So for this example, okay, I just try to copy the experiment. Okay, I prepare 3 potato cylinder, okay, with 5 cm. And after immersion, okay, immerse them in solution A, B, C, okay. So after we put it in solution A, it becomes longer to become X cm. I put it in the solution B, it becomes 3 cm, become shorter. And I put it in the solution C, it becomes 5 cm, unchanged. So that's why in this table, okay, very quick calculation, 5 to 8, okay, 3 cm longer. 5 to 3, 2 cm shorter, and 5 cm to 5 cm, no change. Okay, 0 centimeter, okay, change. So that's why for this part, okay, the most important thing is the net movement of water molecules. So imagine why the potato cylinder will become longer, shorter, or remain unchanged. It's because the water movement, whether they go into the cell, okay, or they go out of the cells. So imagine that from 5 cm to X cm, that means actually the water molecules, there is a net movement going into the cells, because once the water go into the cell, it will make the cell become a bit bigger, a bit turgid. Okay, so every cells, they become bigger, so it will increase the overall length of the potato cylinder, right? And for the case in solution B, okay, it becomes shorter. Okay, so that's why, because the water molecules, there is a net movement of water molecules that go out of the potato cells. So that's why every potato cell will become a bit shrink, a bit smaller. Okay, so that's why the overall length of the potato cylinder will become shorter. And for solution C, okay, there is no change, because there is still movement of water molecules. Some water molecules go in, some water molecules go out, but there is no net movement of water molecules in or out of the cells, okay, of the potato cells. Okay, so at least you get this idea. So I use the solution C as the example, okay, and it's just like the solution Z. So the potato cylinder immersed in the solution C or solution Z is neither longer nor shorter than before. That means remain unchanged. So the concept is that there is no net movement of water molecules in or out of the cells. And it implies that, it shows that actually the water potential of the sucrose solution, it should be the same as the water potential of the potato cell cytoplasm. So that's why there is no net movement of water molecules in or out of the cell, and it will not affect the overall length of the potato cylinder. Okay, so that's why you should know that for Z, okay, it should be in the middle, no change, right? Okay, but one is longer, one is shorter. So which one, the solution, it should have a higher water potential, and then the water, there will be a net movement into the cell to make it become longer. Or which solution, it has a lower water potential than the potato cell cytoplasm, and then make the net movement of water molecules going out of the cells, and finally make the overall length of the potato cylinder become shorter. Okay, so that's question 3. So for answer A or answer C, you tell me the final answer. And then for question 4 to 6, okay, it is a series of questions, okay, so it's talking about the experiment of osmosis. Also, we have a potato and then cut it from the base and then make a cavity, okay, to the potato, and then the potato, okay, for the cavity, we add the 10% sucrose solution. And for the whole potato, okay, we put it to the distilled water, okay, it is the cut surface, okay, and then wait for 1 day. So for this setup, I would like to mention two things first. So the first thing is that potato skin is impermeable to water, okay, so that's why actually there should not be osmosis taking place through the potato skin, okay, because it is impermeable to water. So only the cut surface, okay, will come in contact with the distilled water here. And the same idea is that because in the cavity here, we also have the cut surface here. So that's why this part of the cut surface will be in contact with the 10% sucrose solution. Okay, so that's the basic idea. So that's why the osmosis, it will only take place, okay, in this area and in this area. But for the two sides of the potato skin, it is impermeable to water, so no osmosis will take place. And finally, we have the result. After 1 day, the level of the sucrose solution rose. So that means this level will become higher than before. Okay, become higher than before. So for this experiment, what you need to know, the first thing you need to do is that beware of the concentration difference of the 10% sucrose solution and distilled water. And then for the second step, you can determine the water potential difference between them. Okay, 10% sucrose solution and the distilled water, which one has the higher water potential? And later on, I will use the symbol of the water potential psi, okay, to represent the water potential, to try to save some space for the wordings, for the typing. Okay, so beware of the symbol, okay, psi. So for question 4, is talking about why the level of the water, okay, the sucrose solution will rise. Okay, so the first idea, remember the good practice: distilled water, higher water potential; 10% sucrose solution, lower water potential. By compare both of them, okay, that is the very good practice for those osmosis question. You need to know this first, okay, because somehow the question it will ask you, okay, no matter what. So for A, B, C, D, okay, actually A, B, C are wrong, okay, but why? Okay, I explain to you bit by bit. So I will talk about A, B first, okay. A, B actually, okay, is about the wordings, okay. Water was drawn mainly from the cells in contact with the sucrose solution. Water was initially drawn from the cells in the contact with distilled water. Both A and B cannot describe the whole process, okay, because it should be in a sequence, okay. So while the level of the solution it will rise, okay, by the osmosis, because actually it should be because for the water initially, okay, draw from the cells surrounding the sucrose solution. So the cells here, okay, the water will be drawn, okay, from the cells to the sucrose solution. And then bit by bit they lose water, and then they ask the cell next to them, and next to them. It will be the outside distilled water. So that's why for option B is also wrong, because the water, it should be mainly drawn from the cells in contact with the distilled water, but not initially. So that's why if we change the adverb, okay, A to B, we swap it. So somehow these two statements will become a bit correct. But A and B, they only describe a certain step, the first step and the last step, okay. So that's why it's not a full version. So that's why A and B are also wrong. And for C is wrong because it's talking about cell wall. But for the osmosis, it's talking about the presence of differentially permeable membrane. So that's why C cannot fulfill the criteria, okay. And for D, it's talking about cell membrane. So it's just like this picture, okay, in your book, okay. It shows the 3 criteria of the osmosis: net movement of water molecules across the differentially permeable membrane, and from the region of higher water potential to a region of lower water potential. Okay, so we are going to talk about question 5. So for question 5 is talking about cells here. Okay, the appearance of a cell in contact with distilled water in the beaker at the very beginning of the experiment it looks like this. Okay, so it's a bit easy to understand that because these cells is contact with the distilled water, so that's why actually the cell should be a bit full of water. Okay, just a bit full. So at the end of the experiment, okay, so when the cells keep contact with the distilled water, okay, so what should it be? So the question is that we call the water potential difference between the distilled water and the cytoplasm in the cells. So water potential of the distilled water is the highest, right? So when we compare it with the water potential of the other solution or the cytoplasm, okay, so I think you should know the answer. And after you can determine the water potential difference, and then you can tell me the direction of the net movement of the water molecules. Okay, there should be a net movement of water molecules. Okay, from the distilled water or from the cells to distilled water or to the cells. Okay, the direction from where to where. Okay, so for these two questions in blue color. So if you can also copy these two sentences and with the correct comparison, okay, higher or lower, or the direction from where to where, if you can answer it correctly, I will give you one more bonus mark. Okay, so for the questions, okay, the guiding questions are these two parts. And one more thing I would like to mention is that beware of the size of the vacuole, and vacuole. Okay, so take a look at the size, and then we call the function of the vacuole. It is containing something. So that's why I think you can probably get the answer once the cells keep contact with the distilled water. Okay, so somehow it should be either A or B. Okay, to become a bit turgid. Okay, but never become this plasmolized state. Okay, plasmolized condition. The cell membrane detached from the cell wall. Okay, not these two, but A or B. But A or B is the final answer, you tell me. Okay, so take a look at the size of the vacuole. Okay, which size should be the suitable one once the cells is keep contact with the distilled water, A or B. And number 6 is talking about how can we modify the experiment, okay, to get another new result. So which of the following treatments will lead to a higher final level of the sucrose solution after 1 day? So for the initial level here, and after 1 day, the final level will maybe a bit higher, okay, as what the experiment said, what the result mentioned. So what we are going to do is that, for example, this blue line is the final level, okay, and after the modification, okay, 1, 2, 3, after the modification, I would like to get a higher final level of the sucrose solution. That means this sucrose solution level will become higher, okay, than before. Okay, so clarify the question is asking how to make the final level higher, but not just attain the result quickly. Okay, I wanted to get a higher level. So that's why what we are going to do, okay, we should make the water potential gradient a bit steeper. Okay, a bit steeper. So that's why there's less steep water potential gradient, and it is a steeper water potential gradient. So that means outside it is distilled water. Okay, the water potential must be that high, But the sucrose solution, okay, I would like to make the difference of the water potential between them, okay, become bigger. So option 1, use 5% sucrose solution instead of 10%. So that means we use a dilute sucrose solution, okay, not that concentrate, from 10 to 5%, okay, dilute concentration. And then actually it's not the method, because it will make the final level of the sucrose solution become lower. That's not, we do not want. And then for option 2, use 20% sucrose solution. That means from 10 to 20. So somehow it will make us the steeper water potential gradient. And then finally, actually the final level of the sucrose solution, it will become higher. That's what we need. And at the same time, it will also speed up the osmosis. Although it's not what we want, okay, to speed up the osmosis, but that's the fact, okay. After we use a steeper water potential gradient, okay, at least you know that it will make us the higher final level of sucrose solution, okay, a steeper water potential gradient. And then option 3, peeling off all the potato skin instead of just cutting the bottom. So that means it will increase the surface area, okay, to keep contact with the distilled water. Yes, it's correct. And it will increase the surface area for osmosis. Yes, it's correct. But it's only making the osmosis rate become higher, a faster osmosis. But actually, that's not what we want, okay? Because the solution, okay, is still 10%, and outside is distilled water. The gradient, the water potential gradient, is not becoming steeper than before. So that's why it's same water potential gradient. It will not give us a higher final level. It will just speed up the osmosis. So that's why for option 1, 2, 3, you know what should be the answer. Okay. And for question 11 is that it's talking about phagocytosis. So you can recall the question, okay, recall the answer from the book actually, because you have this diagram. So what I would like to mention is that what is the macrophage? Macrophage, we shall talk about in the body defense chapter. Macro, it means big, because the opposite word is micro, okay, small. And for the suffix phage, that means eating. So that means that macrophage is the white blood cell, okay, which can engulf the bacteria and kill them. Okay, so that's what our body defense system works, right? So for the macrophage, undergoing phagocytosis to engulf the bacteria, okay, actually what does it do? Okay, for the first step, okay, we have 1, 2, 3, 4, but we need to put them in the correct order. So for so-called number 4, the macrophage engulf the bacteria. So that is the first step, okay, because it will form the pseudopodium, okay, to surround the bacteria, surround the target particles, okay. In this case, it is a bacteria, okay, engulf it. So it's going to engulf it, okay. So that's why at least you should know that for B and D, they must be wrong, okay. So only A and C. So A and C, A or C, which one is the correct answer? So we take a look here. So number 4, going to engulf it. So how about 1 and 2? Okay, which one should be the first? Okay, engulfing or engulfed. Okay, it's trapped in the vacuole. Okay, so and then number 3, the bacterium is digested by the M side. That means I kill them. Okay, the cells will kill the bacterium. So it's just like this step in the green box. Okay, so there is some M size is released to the vacuole, fused together to kill the bacterium. Okay, so that's the number 3. So that's somehow, okay, for number 4, okay, and then for number 3, where should be number 3? Okay, at the last or the second last? You tell me the answer. So that's question 11. So that's the part for the MC questions. Remember to answer the bonus question. Okay, so and then we can move on to the part 2 for the long questions. Bye bye.